watching your reaction videos.
No.
We pronounce some
I think someone can. Like, how am I saying that wrong?
It's some.
You know what? Just for that, I'm gonna start calling him Salmon Con.
You still have reaction videos.
If, or when I do reactions about it, so we're doing.
And you can only win.
We got
Hi,
computer.
Nobody gets
Let's go.
in about that. I also do fight recasts, but nobody gets in.
Oh, you did bowling too.
Oh yeah.
I was really interested, but how was
So I think it's kind of easy, right? Like just, I love that scene with that. Somebody got that story. He's just ripping blood everywhere. And I'm like,
it's so like the whole speech. Every 5 seconds is something. That's just weird.
I remember, like, if you've got a 5%, you should check out. Yeah, don't worry about that. That's
Single coconut.
I think it was. I mean, I didn't pay money to see it. I didn't pay any money to see it.
You don't want to see the story.
Have you ever seen that?
You realize how much that's not how it's story.
I have no doubt of that. Have you ever seen that classic movie on BDLJ? I know it is. I know it is.
Oh, you have to go.
It's great. The end at the train station where they're having the fight scene. Oh my god, that's so bad. Shut is strong. I'm like, this is terrible. Oh yeah, it's like a foot away from Yeah.
The story is more.
Oh yeah, I know. I mean, like, it's the best 3 hour epic about, you know, going back in forever.
You must look at it. She can't say that.
I'll choose sometimes. Oh my god, that'd be like a 6 hour movie then. No, I'm not.
Every like line plays positive.
Yeah.
Play it.
I've done yeah, I've done that before. I had to do that with Sultan, but that was not fun.
I was American one. It's kind of similar to that, but it's wrestling.
You better remember that.
I watched all American. They come out like once a year.
Oh,
He's like the equivalent of.
I mean, like, literally, like, you don't know.
Okay.
It's like people camp out outside of their house. Yeah, see them come Oh, yeah, that's the one. That's the
Oh, Francis, right.
I really love it.
Okay, let's get started. So, just to be clear, this is not on your exam on Friday, but it will be on your finals course at the end of the semester. And does everybody have something with Poll Everywhere capabilities today? Because we're going to have a lot of participation through that today. So today we're going to start our talks on metabolism, starting off with sugar, with monosaccharides, particularly glucose, with glycolysis. And the little cartoon here is just a screenshot from the Oprah Winfrey Show from back in the day. Might be before your time, I'm not sure. But we're going to be following those phosphates. Phosphates are going to be flying everywhere. We're going to look at where they're going, how they change the energy, and how they change the molecule until finally the glucose molecule that we start off with is no more and is very small, broken down into a little fragment of what it started as. So today we're going to just give an overview of glycolysis and then go over the 2 different phases, the investment phase and the payoff phase. And then we're going to talk about how we need to regenerate NAD because we use NAD in glycolysis. And if you just go through glycolysis without regenerating the NAD, eventually you're going to use that all up and you're not going to have any more. So for today's learning objectives, we'll describe glucose's entry into the cell and describe the metabolic pathway that leads from glucose to pyruvate. So this is done in 2 phases. We'll take note of where the ATP is consumed and where ATP is formed. And then there are only going to be a few key enzymes that you'll have to know. These are all starred on the slide, and I'll make sure that I emphasize this is one of those enzymes, and know what the substrate, the product, and basically what it does and why it does it. And also, of course, why it's important. And then the entry point of other compounds into glycolysis, we'll also be talking about that, as well as we'll describe what happens to pyruvate at the very end. There's 3 typical fates, and so we'll talk about what goes on there and why those 3 fates exist and what predominates. So coming to our metabolism slide, as I promised, we'll be looking at this almost every day that we talk about metabolism. Today we're going to talk about this little section here in red, where we're going to be going from glucose down to pyruvate. So just as a review, remember metabolism is a balance between catabolism and anabolism, where catabolism is we're degrading our complex molecules. We're starting with our polysaccharides, and we're going down to our monosaccharides, and then further down into pyruvate, and eventually we're going to cause releasing of energy. Whereas anabolism, you're constructing molecules. We'll talk a little bit about this here and there, but for now, today we're going to be talking more about that degradation. And so we're going to, again, be focusing today on these polysaccharides, going through, turning into glucose, going into acetyl-CoA, and eventually going through oxidative phosphorylation, producing ATP. And the reason why we start with glycolysis is that it's one of those feeder pathways where a lot of things can feed directly into something directly into glycolysis or right after glycolysis. So if we learn the main pathway first, making all those connections where they come through will be a lot easier. So we'll start with just a general overview of glycolysis. So glucose is the major carbohydrate available for use by cells and is rich in free energy. So if you were to look at the complete oxidation of glucose, we have glucose plus oxygen. We go to carbon dioxide and water. You're going to release around 28,000 kilojoules of energy per mole of glucose. That is a lot. And remember, when we break ATP, we're releasing only 30 kilojoules of energy. So this is multiple times the amount of energy released when you break down a glucose into carbon dioxide than you would release from just a single ATP. Of course, the whole point of breaking down this glucose isn't to form one ATP. We're going to be forming multiples of these. So that we're going to try and harness as much of this 2,800 kilojoules of energy as we can into building the ATP. So glucose has a lot of different choices once it's inside of our body. It all depends on what our body needs and how it gets used. It can be used to make structural molecules for the extracellular matrix. It can use to be put into storage. If you have a high caloric intake, you don't need to burn all of that sugar at once. We're going to store some for leaner times. And so our body makes glycogen, or if you're a plant, you'd make starch or some other type of sugar to store that. Glucose, however, if you need energy, and most people do on an everyday basis, you're going to break it down and make pyruvate and do glycolysis. Alternatively, if there's a lot of oxidative stress, we use something called the pentose phosphate pathway to make ribose 5-phosphate. And we'll talk about this a lot later in the course after we talk about all of metabolism going from sugar to ATP with oxphos. Yes?
I could be wrong, but is this recording? Usually there's a sign that says recording.
It should be, yeah. Let's just double check. Yeah, it says it's recording. That's all right. So it's always good to check so that we have one for later. All right, so we have all these different options for glucose inside of our body. All depend on what our body needs and how we will therefore proceed. Let me just get that laser pointer back. All right, so glucose will enter our body through specific transporters. there are multiple versions of these, which all are expressed either basally in different types of tissues. So you can see some of these are only in the liver and pancreas, whereas some are in only muscle and fat. Some are in every single tissue. Some are just at low concentrations. Some are at high concentrations. For now, just concentrate on the actual mechanism. We're not really too concerned about all these locations as of yet. The way in which the glucose transporter works is that there's some extracellular signal. Typically, it's the insulin signaling pathway that activates its receptor and sends a signal transduction cascade through the cell. And inside the cell, there are these glucose transporters that are held in intracellular vesicles. Now, these vesicles are held inside of that cytoplasm. Only on need basis will it be brought to the surface. So once you have that cell signaling cascade, that is the signal that we need to move these transporters up to the cell surface, to that cell membrane. And then there'll be an exocytosis, so this vesicle will fuse with that cell membrane, and the transporters will be active, which will then allow the glucose to flood inside of that cell and just enter in. Of course, there's also another pathway which isn't shown here, which would then take those glucose receptors off the cell. Once you have enough glucose and the cells don't need it anymore, they're going to remove those transporters from the surface and bring them back in through vesicles as well. So the process just works the same, but in reverse. So there are two phases of glycolysis. And so the input at the very beginning is glucose, and then the basic overview is you're going to invest 2 ATPs. So you're going to hydrolyze 2 ATPs to form ADP during what is called the investment phase. So invest your ATP, investment phase. And then you will go through various steps where you then will create 4 ATPs and 2 NADHs. And this is the payoff phase. So this is where you're actually building your ATP storage of that energy. And at the very end, you'll produce pyruvate. Yes?
Have glucose ever invest and never get the payback?
I'm sorry?
You like have glucose ever invest to ATP and never get payback?
So the payback comes later. Yes, right here, we get the 2 ATPs broken down. We get 4 ATPs being built. So net, well, at the very end, we get 2 ATPs. We also will get those 2 NADHs, and we form these 2 pyruvates. So you were starting with a 6-carbon sugar, and then we're ending with 2 pyruvates. Yes?
Is the payoff phase the yellow block?
Yes, the payoff is the yellow block.
Okay.
Yeah. So energy generation or payoff, it's synonymous. So we'll be going through these step by step. Yes.
The NADH does it give off?
So not at this point. So at this point, we're just forming NADHs. We come later when we get to oxidase phosphorylation. All of those electrons that are stored in that NADH will go through the electron transport chain and then eventually form ATP at the end. But at this point in glycolysis, if we were to stop right here, we just formed NADHs, and they're doing nothing else other than existing. And we'll talk about how that can happen and where you actually don't go through to the rest of metabolism, where you kind of just stay cycling at glucose with glycolysis. But that's later in the talk, and we'll highlight that. So the whole idea is that you're going to put in energy to make more energy. As you can see, you're putting in 2, but you get out 4. So the net is 2. So you do get extra energy out of this process. So just as an overview, you're starting with a 6-carbon sugar. You go through glycolysis. You end up with this 3-carbon molecule called pyruvate. The structure of pyruvate is not important for you guys to memorize. However, just familiarize yourself with it, because we're going to be seeing this molecule quite a lot and just seeing how things, how different parts of the molecule through organic chemistry are changing. Now, once you have pyruvate, you can go to 3 different options. You can make ethanol, lactate, or also called lactic acid, or you can completely oxidize it with oxygen to make carbon dioxide and water. this is the pathway through this complete oxidation where you're going to get the most energy out of it, and this is what is involved with the mitochondria. However, there are situations in which we just can't do it, and we'll talk about why we go into lactate or ethanol again later in the talk.
So complete oxidation has the most energy comes out of that.
Right. So complete oxidation, you're going to get the most payoff from glycolysis, from that pyruvate. You're going to get way more ATP than with the other fates of pyruvate.
More buck for your bag.
Exactly. So the reactions of glycolysis can be characterized based on what is actually happening. So there's 3 different processes that are occurring. A lot of times we're going to be adding phosphoryl groups to the glucose. This yields compounds with low phosphate group transfer potential priming. And so we'll talk about what this means, but as we saw from the cartoon at the beginning, we're moving phosphates, we're adding phosphates. Phosphates are moving around a lot, and it's to make different types of energy rich or energy poor compounds. Then we will chemically convert these low phosphate groups. So we transfer potential intermediates into compounds with high phosphate group potential. And then we're going to chemically couple the energy. So we'll hydrolyze these phosphate groups, transfer potential compounds to the synthesis of ATP, and transfer phosphate groups to ADP. So we are going to form ATP, and we're going to be essentially collecting all of this energy that we've been transferring around. So this little flow diagram is very similar to the one that we saw before, except here it shows a little bit more of the key metabolites in between in the phases of glycolysis. We're going to go through all of these steps step by step in the next couple slides, but for now, this is a good slide to look at to try and familiarize yourself where the ATP is being consumed, where it has been utilized, and just general flow of the entire process. So it's a good review slide. So glycolysis, starting off with the investment phase. So this is the first 5 steps of the reaction. This is the energy investment phase. Glycolysis will start again with this glucose. You'll invest your first ATP to produce different types of intermediates, and eventually you'll actually split that glucose into 2 3-carbon sugars, some of which we are very familiar with, like dihydroxyacetone and glyceraldehyde phosphate. So we're essentially just breaking that sugar and cutting it in half. And that's the whole point of this first investment phase, is to break glucose into 2 pieces. So the first step is one of those very important enzymes that I told you about. It's here, starred. This first step is done through the enzyme called hexokinase. So this is the first ATP investment. So you start off with glucose, you have your ATP, that ATP gets hydrolyzed, and the phosphate from that ATP is transferred directly to glucose. So that phosphate, usually when we think about ATP being hydrolyzed, it's ADP and then a free phosphate. This time the phosphate is directly going onto that glucose. And so this is the very beginning. It's ATP dependent, so if you are low on ATP, if you don't have any ATP, you really can't proceed at this point. You're just going to be stuck with glucose. You're not going to go much further. The initial reaction serves to put a low energy phosphate onto glucose. So this is a very low energy rich molecule. So if we go back to this chart that we looked at last time on Monday, we can see ATP is here in the middle. Glucose 6-phosphate, which is the product of hexokinase's work, is very low in energy. So it's about negative 13.8 kilojoules per mole, which in relationship to ATP being at negative 30, it is much lower. So this, however, has a very important step in that adding this phosphate onto glucose makes it so that glucose cannot leave the cell. So once glucose is in a cell, it can move in and out across that cell membrane through different transporters. But once it has that phosphate group on it, it is stuck inside that cell. The transporters cannot recognize it, and so it's not going anywhere. It is stuck there. The next phase is done by an enzyme called glucose 6-phosphate isomerase. So it's an isomerase, so that just means we're moving and we're changing the structure of our substrate. So it's an isomerization of that glucose 6-phosphate. So this enzyme we'll isomerize the aldose glucose 6-phosphate to the corresponding ketose, which is fructose 6-phosphate. So if we look here, the aldehyde group on the glucose 6-phosphate gets moved, and we produce a ketone group, which is right here. So we're just moving that oxygen 1 carbon down on that chain. So transferring the carbonyl oxygen from carbon 1 to carbon 2 has an important effect. The hydroxyl group generated at carbon 1 can be readily phosphorylated in the next step. So this is, even though it's not one of the starred ones you have to memorize, it is a very important step, because the next step is one of those very important ones. So it prepares us for phosphofructokinase. So this enzyme will take that fructose 6-phosphate, which was the product of the last enzyme. We add another phosphate group here. So the phosphate here in blue is the phosphate from the original ATP hydrolysis by hexokinase. We're now going to add another phosphate group to it on this other side, and that's going to be pink here to make fructose 1,6-bisphosphate. So bis here is just a term that is used very similar to di, just means 2. So fructose 1,6-bisphosphate. So this catalyzes this is the second ATP investment step. So here is a second ATP being used to give this hexose derivative, which is phosphorylated on both sides. So it's we have 2 phosphoryl groups on it now. This sets the sugar up for a symmetric split down the middle. As you can see, if you split it down the middle, it's pretty symmetrical, relatively. Now the products can be converted from low energy to a high energy molecule. The phosphofructokinase reaction is the primary step at which glycolysis is regulated. This is often considered the commitment step. And the reason why it's the commitment step is that once you are here, once this enzyme is active, there is no turning backwards. You have to go forward with glycolysis. Previous to this, the hexokinase and the enzyme before here, they are involved in other pathways as well, so you're not locked into glycolysis yet. But once phosphofructokinase works on the molecule, it is dedicated to going through glycolysis. So as you can see, this reaction is very heavily skewed going forward. It doesn't really go backwards. That equilibrium is not really present. Step 4 is where we actually get the cleavage of that 6-carbon sugar into 2 3-carbon sugars. This is done through a enzyme called aldolase. So now we're going to form 2 triose phosphates. So we started with a hexose, and now we're getting 2 trioses. The fructose 1,6-phosphate aldolase catalyzes the splitting of the sugar, and this is this helps give the name glycolysis. So the lysis. Glucose being split, basically. The 6-carbon sugar, fructose 1,6-bisphosphate, is cleaved to give 2 3-carbon intermediates, glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. So these are just your simple trioses with an added phosphate group on them. Aldolase cleaves the fructose 1,6-phosphate under intracellular conditions, even though the equilibrium lies far forward towards the fructose 1,6-bisphosphate under standard conditions. It will continue to go forward to try and push to continue the glycolysis. Remember, the phosphofructokinase is that committed step. Once you have that, you're going to be going forward in glycolysis. So even though this is a much more stable molecule, we're going to try and push it forward and try to get to the end of glycolysis. So the last phase of the investment phase is done by an enzyme called triose phosphate isomerase. And this will only work on one of those trioses. So you remember that we form 2 trioses. We form a dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. that glyceraldehyde 3-phosphate that forms is just going to go on to step 6. However, that dihydroxyacetone phosphate is going to be converted into another glyceraldehyde 3-phosphate. So that at the end of this investment phase, you're going to end up with 2 of these glyceraldehyde 3-phosphates. So this is an isomerase. We're just moving different parts of the molecule around to try and get 2 exact same molecules. So we have this isomerization event here. The pink, you can see where these hydrogens are moving, and the carbonyl oxygen is moving as well. So the dihydroxyacetone phosphate is one of those ketoses. So then when this triphosphate isomerase comes in, it will then move that carbonyl carbon to make it that aldehyde, and you end up with the glyceraldehyde 3-phosphate. Now, glyceraldehyde 3-phosphate is also one of those sugar phosphates that we talked about as far as having energy, and this is one of the lowest. So here you can see that there's pretty low amount of energy here. There's only 9.2 kilojoules of energy, which again is a factor 3-fold lower than that you'd get after splitting ATP. So we started with glucose, which was a 6-carbon sugar. We added some phosphates to it, and then we broke it into 2 3-carbon trioses, both of which have phosphate groups on them, but now they're both very low in energy. So we've invested energy, and we've created things that are very low in energy. It seems a little counterintuitive that you're going to be making things that are so low in energy, and you're going to be putting in energy. How are we actually going to get any of this energy back out and actually make more energy than we started with? So that's where the next phase comes in. At the end of the phosphate, this investment phase, if you were to stop here, you really didn't do much. So let's just have a quick review question. So in the preparatory phase, this investment phase, we call it, this energy is produced for the cell to use. True or false? Everyone respond? No? No? Keep moving. It's like watching the polls last night. Is it going to stop? All right. So the majority have false. And that is the correct answer. So at this point, we've only put in energy. We've not gone any further than that. We've made things that are actually very low in energy, and we now have to figure out some way to get all that energy back. We need to make more, because I told you at the very beginning, we're actually going to be making 4 ATP. We put 2 in, so our net is going to be 2. We're going to be better off than when we started, but somewhere that has to come back.
So far, what you just talked about is just investment phase, right?
Mm-hmm.
So that's why the answer is false, because it's just investing.
Right, we're just preparing and investing, yeah. We're not actually creating anything yet. Other than we made two trioses. Phosphates, but those things are very low in energy. They're not really an energy source for us. So our payoff phase is the last 5 reactions of glycolysis. This is where we're going to actually produce our energy, and it's either called the payoff phase or the energy generation phase, something to that effect, where you understand that we're making ATP. So the 6th step, we have glyceraldehyde 3-phosphate dehydrogenase coming in. We take our glyceraldehyde 3-phosphate, and here you can see that NAD is one of its cofactors, and we're going to then make NADH, so we're transferring these electrons, and then we end up with 1,3-bisphosphoglycerate. So we have this phosphate group being transferred onto this molecule. This is an anhydride linkage, which actually does contain some good amount of energy, and we'll see where that is on the next slide. So this is the first intermediate with a high phosphate transfer potential, and we we get this activated carrier being produced with electron transfer. We have the NAD plus going to NADH. So NADH, as I mentioned earlier, will then later be shunted into oxidative phosphorylation to produce ATP. But for now, if we were not to have oxidative phosphorylation going on, if we were just at the end of glycolysis, all we have is NADH. So this 1,3-bisphosphoglycerate, again, is a very high energy phosphate. So if we look at our chart that we had from last time, this 1,3-bisphosphoglycerate is actually much higher than ATP. So ATP is around negative 30.5 kilojoules. Here we're up at 49.4. So we're significantly higher in energy. So So remember, we're starting with things that are very low in energy. We're starting down here with this glycerol 3-phosphate, and yet we're ending up here with this 1,3-bisphosphoglycerate. So this, again, is due to that coupling of those activated carriers with this enzyme, so that we have a coupling of an exergonic with an endergonic reaction. We can transfer this energy and create something much more higher in energy and higher energy richness than what we started with. And so you had this aldehyde group being oxidized, which was the exergonic, and then we also had this acidic group, which was phosphorylated, which was the endergonic reaction. Those two came together, and we created this high-rich energy molecule, this 1,3-bisphosphoglycerate. So now that we have this 1,3-bisphosphoglycerate, remember we have two of these because this happened with each of those glyceraldehyde phosphates. We had started with two of them at the beginning of this phase. So everything from this point on, you have to remember, is doubled. And so phosphoglycerate kinase will come in, and this is the first substrate level phosphorylation. And here you can see that we're going to form our ATP. So we have this two phosphates here. One is white and one is yellow. The yellow one is actually then going to be transferred to the ADP, and we produce ATP. And we're also going to release some energy in the form of heat, but that energy is just going out and is essentially lost. We can't harness any more out of that. And so we end up with 3-phosphoglycerate, which only has now one phosphate on it, where we started with the two. We now have one phosphate. And so this is the first formation of ATP. And this is done by this phosphoglycerate kinase. So usually when you hear the word kinase, you think of something where that adds a phosphate. Here you see it's actually taking off the phosphate. And this is an interesting case of where the activity of the enzyme was actually not really fully appreciated when it was discovered. And so when it was discovered in the test tube, they saw that this enzyme could add a phosphate. However, it was an artificial situation where in actual physiological conditions, it's removing that phosphate. So it's one of those types of enzymes where the name actually doesn't quite make sense, where most of the time enzyme names do make sense. This is just one of those rare cases.
Wait, so you say kinase is adding phosphate, but phosphate glycerate kinase is not adding a phosphate?
Right, so kinases usually add a phosphate onto things. And that's just how they work, is they usually add a phosphate. In this example, it's actually removing that phosphate. So removing the phosphates are usually done by enzymes called phosphatases. And so a better name for this would be phosphoglycerate phosphatase. However, because it was already named and already characterized in the test tube before they realized what it was really doing in our cells, they couldn't change the name. So the name kind of stuck, even though it really doesn't quite make that much sense.
Does this have anything to do with body heat? Does this contribute to the body?
There's some contribution to this. We're going to talk more about body heat a little bit later when we talk about mitochondria, but there is some contribution because, again, that is some free energy just escaping as heat. And we do a lot of glycolysis. Glycolysis goes on all the time in our body. So the next step, we're going to take our product from the last one, 3-phosphoglycerate, and then we're going to just basically move that phosphate group up a carbon. So it starts at 3-phosphoglycerate and now it ends up as 2-phosphoglycerate. So this is a rearrangement of the intermediate. Not that much interesting with that step. But here we have an enolase, step 9. Here we're taking that 2-phosphoglycerate and forming the most energy-rich compound that we have in our body, the phosphophenol pyruvate. So here you can see it is about twice as energy-rich as ATP. So ATP is around -30.5. This is at -61.9 kilojoules per mole. So twice the energy richness of an ATP. This is among the most important intermediates in glycolysis and generates an intermediate with the highest phosphate transfer potential. So then that brings us to our last step, which is also one of the starred ones. This is our final formation of ATP. This is also one of those enzymes where it's removing that phosphate group, even though it's called a kinase, like what happened before. This is pyruvate kinase. We are removing that phosphate group, adding it onto ADP to make ATP, and we get pyruvate. Now, pyruvate can take a couple different forms because there's resonance with all of those double bonds that are floating around in there. Again, I said don't memorize the structure, so it doesn't really matter. But this double bond is critical for the phosphoenolpyruvate resonance destabilization. So because there's so many double bonds, there's some destabilization forces going on there. And the negative charge from the phosphate and the carboxylic acids do repel each other, which helps with that removal because this is so high energy rich. Removing that phosphate could be difficult, but because of those negative charge repulsions, we're going to actually be able to take it off much more easier with this enzyme to add it onto that ATP. So the pair of electrons do typically want to be on this oxygen because it's more electronegative. It will repulse that phosphate away, and then it will release energy, which is captured in ATP. Interestingly, unlike the other phosphoenolpyruvate kinase, this pyruvate kinase does not release any excess energy as heat. This is all the energy goes directly into that ATP formation. The reason why this one is important, not only is it the final step in glycolysis, we're producing ATP, but we're also producing that pyruvate. Pyruvate is the ultimate goal at the end of glycolysis, is to get your ATP out and then to form pyruvate. Having that pyruvate then lets us do multiple other options, and it's a very utilizable molecule. It's not that we're producing some type of waste molecule at the end of glycolysis. This is something that we can use very, very easily. So as a little review question, what is the net outcome per molecule of glucose? Okay. So we have a little bit more spread. Everyone had a chance to answer? Doesn't look to be moving anymore. So totally, we're going to be producing 2 NADHs, 4 ATPs, and 2 pyruvates. This is all, yes?
So are we not counting the energy investment phase, the 2 ATP that are eaten?
Right, so 2 are eaten. 2 are eaten, yes. Yeah, not literally eaten. Yes, 2 are consumed at the beginning, and then we'll get 4 out at the end. And again,
but since it adds for the net,
it should be the 2 there, yes.
so
Yes,
that's why.
that is, yep, that's a mistake on my part. Yep, so it should be 2. because it does say net. If it said total, it would have been 4. Yes. Very good. 4 were produced, but only 2 outweighs what you put in.
So for this one, it would be C, technically.
Was that,
If
yes,
you're looking at,
C.
okay,
If you're looking at this, yeah.
the net, but the total would be B.
Total is B, net is C.
Okay.
Correct. So we have this phosphofructokinase step of glycolysis. Why is this called the first committed step? Okay, just keeps going back and forth, back and forth. Everybody have a chance? All right, it looks like it's stopped. So why is it called the first committed step? So is it the first step of the pathway? No, so that's not choice A. Is it the last choice of, is it the last step in the investment phase? So does it bridge that hexose triose phases? So it's not choice D, no. Is it the only one that's reversible? Irreversible, sorry, irreversible. Is it the only one that's irreversible?
Are all the steps past that irreversible?
Mostly, yep.
Mostly.
Yeah.
Okay, so that's confusing.
Well, there's the, yeah, so they're pretty much irreversible. Yeah, there's, the reason why I say that is because when we get to gluconeogenesis, some of them are in both pathways. So there are conditions in which you can go backwards.
Yeah, biological lexicons figure it out.
Well, you know, it's all about utilizing the same thing so that you're not making more enzymes, you're using the same to do 2 different things. So it's choice C. So as the entry point, so it's for substances that can bypass the hexokinase and glucose kinase. And as we said, hexokinase and glucose 6-phosphate isomerase, those first 2 steps, once they produce their products, those can also be used in other pathways as well. So once we get to the phosphofructokinase, that the product that it produces can only go forward. It's not going to be used in any other pathway. You're going to be locked into glycolysis. So now that we've made pyruvate, so we've broke our glucose down, we split that out into our trioses, we now form pyruvate. We've produced a lot of NADH. And so if you were to continue with glycolysis over and over again and not regenerate NADH, you'd eventually run out of NAD, and without that NAD, you couldn't do any more glycolysis. So you're kind of stuck. You need some way to create more NAD so you can do more glycolysis, so you can get more ATP out, and the cycle will continue. So our bodies have made 3 different pathways in which we can actually generate this NAD. So from pyruvate, we can either produce acetaldehyde and ethanol, we can produce lactate, or we can produce acetyl-CoA, which further oxidizes down through oxidative phosphorylation, we get ATP and we'll regenerate our NAD. So here you can see in each step, whether it's immediate or it's a couple different steps later, we take our NADH and then we regenerate back to NAD+. So the goal is always to make the NAD+. So fermentation is something that goes on in our body. We don't really typically think of fermentation happening in our own body. Usually we think of fermentation when we think of producing beer or wine or even yeast and bread, doing that kind of fermentation. But our body does do ferment things, and one of which is pyruvate. So this, we take our energy yielding metabolic pathway. We have no net change in the oxidation state of the products compared to the substrates. And this usually happens under anaerobic conditions. So anaerobic glycolysis leads to pyruvate. Pyruvate is then reduced, and so no net oxidation of glucose will occur. So we'll have pyruvate in our NADH and a free proton. And then through the action of the enzyme called lactate dehydrogenase, we form lactate, also termed lactic acid. However, this is a dead-end metabolite. We can't really do anything else with it. However, you can see we have NAD+. So we've regenerated our NAD, and that's the whole ultimate goal. We do release some energy here in the form of heat, around 25 kilojoules. So in this regard, we're just trying to regenerate our NAD, even though we're building up this lactic acid. And so this can produce this cycle of producing, we're going to produce NAD to help run our glycolysis. That the glycolysis produces NADH. We then will cycle it back and forth between this cycling. Now this cycling can occur either with producing pyruvate into lactate or pyruvate into ethanol. And so we'll talk about the ethanol pathway next. But this whole idea is that we cycle this NAD so that we can continue glycolysis. Now this is due to having a limited pool of NAD and NADH. This NAD and NADH does come from vitamins in our diet. It's made. However, we keep it at a certain level. But if it's a limited pool, we don't really upregulate and produce more in times of stress. We typically have this constant pool. So we're going to keep it constant. We have to then regenerate it. So with the other option is alcoholic fermentation. This can also occur inside of the human body, although there are certain instances where lactic acid would form over ethanol. Certain tissues prefer to go the lactic acid route versus the ethanol route. And we'll talk about that in the next slide. But this, we take our pyruvate, we then will remove a carbon dioxide, produce acetaldehyde. We still have our NADH, this comes in. Now we have alcohol dehydrogenase, and we produce ethanol. So this usually will happen in obligate anaerobes that cannot survive in the presence of highly reactive oxygen, such as things that live in soil, water, our skin, also our guts. So a lot of our microbes in our microbiome perform this. However, we do this when animals perform bursts of intense activity, so you need energy and ATP faster than it can actually be produced. So one example are things like Olympic sprinters. Here we have Usain Bolt and all of these other people who don't win. And so what happens are we have, under normal conditions, our pyruvate would then go into the mitochondria, be reduced to be oxidized further, we get carbon dioxide out, and we have ATP to feed our muscles as it's doing those contractions to go help with the running. However, under low oxygen conditions, such as lots of strenuous exercise, these sprints, we're going to be shunting it over to lactic acid. In this way, you can produce the energy quickly. You can go through glycolysis, produce your ATP, and regenerate more of the NAD to continue on with the glycolysis. The process of glycolysis is very quick. Even though there's 10 reactions in total, it goes by very, very quickly. So that when you turn over that NAD H into NAD, you can do more glycolysis and help feed those muscles that ATP that they need for that strenuous exercise. However, if you go through the oxidative phosphorylation pathway through the mitochondria, it's much slower and it cannot keep up with that intense burst of energy. Additionally, when you're exercising and being under a lot of the stress, you have a lower oxygen intake and lower oxygen richness getting into those muscles, so they physically can't do this oxidative phosphorylation, which requires oxygen in the mitochondria. Did I see a question?
Yeah. So when we're doing oxidation, glucose or fermentation or lactate, we have to go through the investment phase and payoff phase before we go through this.
Right. So all of that regeneration of NAD happens once you've already had pyruvate. So glycolysis is essentially over at that point. So you have that end product pyruvate, and then you just have to decide what you're going to do with it. Your body will take in cues whether there's enough oxygen or there's no oxygen, and then try to figure out a way to use that pyruvate to regenerate that NAD and to continue on to create more ATP through glycolysis or further down in the pathway through oxidative phosphorylation. So our last question, in the absence of oxygen, what is the net number of ATP produced during glycolysis when we begin with 1 molecule of glucose? Huh. Yeah, well, that's a typo. Yep, so both answers are right, yeah. So they're both the same, you know? Yeah, this poll everywhere was put together a little too quick, wasn't it? So I guess the answer is 2. So yeah, that's interesting. So yeah, no matter what the oxygen level is, glycolysis will go through either under aerobic or anaerobic conditions. You always will get those 4 ATP forms minus the 2 that you put in there. You get 2 net. So whether there's oxygen there or not doesn't really matter. It's after, and you have pyruvate, and you need to know what you're going to be doing with that pyruvate. That's when your oxygen sensing really comes into play.
That's a question. How come I can't get drunk
From glucose? So how come you don't get drunk from glucose? Haven't heard that one before, I have to say. We don't produce a lot of that alcohol. It's not a way in which we'd actually harness it to get drunk. Yeah, I've not heard that one before. That's good. And not all of our cells are doing this. Our muscles, which are a lot of our body mass, is actually going through that lactic acid pathway preferentially. That acetaldehyde forming into glucose, that happens a lot in like the liver because they express those enzymes so richly. Of course, the liver also has those enzymes which break down alcohol very easily as well. So it doesn't stay around very long. So to summarize, just have this little balance sheet. We have glucose yields 2 ATP of 2 ATP per glucose. It's a very fast reaction, but it only releases a small fraction of what is really available if you were to burn all of that glucose. Fermentation, either into alcohol or lactic acid, is an exergonic process coupled to endergonic process, and again, you're only producing the ATP in glycolysis. The fermentation doesn't add too much. This kind of gives a little idea of where we start as far as energy richness here with glucose and where we end up with pyruvate here at the bottom. And all of these blue arrows are those 3 enzymes that I starred for you: hexokinase, phosphofructokinase, pyruvate kinase. That's either where ATP is being consumed or ATP is being produced, or where there's these big gaps in energy being transferred between something high in energy going to something low in energy. And so next time we'll continue to talk about glycolysis in the context of starting materials that aren't glucose. And then we'll move on to how we can actually make glucose in our body, which is essentially glycolysis in reverse.
So are these the abbreviations and names you want us to know?
Right, so those blue arrows are those 3 starred enzymes. In between, you don't need to memorize those enzymes or those intermediates. No.
All those enzymes, everything, with those big fancy names. You have to.
Just appreciate what's going on in that
I
process.
appreciate it.
Believe me, if I had you memorize every single enzyme that we talk about, you would not be happy.